Hello and welcome. My name is Ethan Gevedon. I'll be presenting my Spring 2021 CURS Symposium project. Our title today is The Synthesis of Selective Pyrophosphate Sensing in Multi-Analyte Material. And I worked under my faculty mentor, Dr. Paul Lanzenbacher with the Department of Chemistry. So as a brief introduction, organic phosphate anions are ubiquitous in nature. They're present in agriculture as fertilizers for many plants, raw materials in industry, and back to nature. Many biological molecules and complexes use the pyrophosphate in various forms. So our focus is around pyrophosphate, or abbreviated PPI for the remainder of our presentation. It plays a key role in many aspects of nature. And one of these processes is the generation of nucleic acids, where it serves as a byproduct of replication. And we have presented the structural formula and a space-filling model of the pyrophosphate anion. Going into our focus, the polymerase chain reaction is a process to synthesize targeted segments of nucleic acid, such as DNA, which are highly useful for genetic analyses. So we can create a primer that is selective for one gene or one DNA sequence out of an entire genome of billions of base pairs. So it's a pivotal technology used in genomics and other areas, almost ubiquitous in every area of biology. And to further this, quantitative PCR allows for the detection and quantification of that DNA replication to occur in real time. So finally, DNA replication, such as in PCR, generates DNA and produces pyrophosphate as a byproduct in equal proportions. And due to the accurate proportionality in production of PPI, we propose a molecular sensor that can use pyrophosphate as an analyte to determine the amount of DNA replication taking place with a higher degree of accuracy. So to further this, currently every method used for qPCR utilizes a fluorescent dye that binds to DNA. So these are usually highly expensive, they're selective, they can potentially be selective, and the cost really precludes widespread use. Another issue is accuracy. With varying DNAs, or with varying nucleotide sequences, the affinity for the fluorescent sensors are often variable, and that increases inaccuracy in the quantization of DNA replication and increases probability of error and false positives, which we look to improve upon. Currently there are no qPCR sensors that utilize PPI as a side product. And therefore, we have a high need for these turn-on fluorescent sensors in multi-phosphate solutions. So we want to create a molecule that is selective for pyrophosphate over other nucleotide sources, such as in qPCR and DNA replication, the nucleotide triphosphates that get incorporated, because those could be in interfering species. So our overall goal of this project, again, is the synthesis of a sensor that is selective for pyrophosphate and produces an amplified fluorescence response in the presence of other phosphate anions. So it's selective over those to provide the highest accuracy possible. And then we have a slight scheme. So the sensor will be non-fluorescent, and then upon addition of the pyrophosphate anion, it will become fluorescent in direct proportion to that addition of the analyte. So here is our proposed molecular sensor. That is the end goal of our project. So there are several functional groups present on the molecule that are described by the different colors. The blue group is a dipicoylamine, abbreviated DPA, which upon addition of zinc nitrate, a salt, the zinc will coordinate to the DPA group, and we will get into the mechanism of the sensor's function in a little bit. The black carbons provide correct spacing to allow correct conformations of the changes in fluorescence and non-fluorescent fluorescence to fluorescent response, and that provides correct confirmation. The green aeroboronic acid elicits binding of the fluorophore upon addition of pyrophosphate, which aids in response. And finally, any red cachetol dye, as we have described here, or red cachetol fluorophore dye, will allow for correct fluorescence response to be seen. To further discuss our goals, the synthesis of the precursor 8 located below was the target of my research this semester. So starting from the scheme below, or the scheme will describe the overall synthesis of the final precursor 8, and I'll discuss them in brief to summarize. We will also discuss each reaction in further detail, along with outcomes of those further in the presentation. So commercial 4-amino 1-butanol was protected with Bocane hydride to produce the carbamate group in intermediate 2. The series of steps leading up to synthesis 6 required a series of protection and deprotection group or reactions to effectively oxidize the alcohol to an aldehyde under conditions that were favorable for us. From intermediate 2, we use the imidazole base and tetrabutyl dimethyl silyl to protect the primary alcohol. This allowed us to use the strong base N-butyl lithium to deprotonate the hydrogen on the primary carbamate group. From there, another addition of Bocane hydride was allowed to diprotect the amine. From there, we were able to deprotect the primary alcohol with terbutyl ammonium fluoride. This produced the diprotected amine with a free OH group. This intermediate 5 was then oxidized with pyridinium chlorochromate over sea light to produce the intermediate 6, which was our target aldehyde. The target aldehyde was then reacted with a second molecule, which we produced during the CURS, or during the spring semester of 2020 before COVID-19, which was reacted with 6 under a reductive amination with the sodium triacetoxyborohydride reducing agent. This yielded the intermediate 7 as one collected molecule. From there, commercially available 2-bromomethyl phenylboronic acid was used to bind to the secondary amine to yield our final key intermediate 8, which we will discuss its importance later. Now going to the mechanism of how the overall molecular sensor works. So, as our proposed from earlier, our proposed structure, we add zinc nitrate, which the zinc ion or 2+ ion will bind to the dipicoylamine. This has strong affinity for zinc. This will induce a conformational change in which the cachetol or catechol group on the fluorophore will bind to the zinc. This effectively sequesters nearly all fluorescence of the sensor, and from there, upon addition of pyrophosphate, the catechol group will shuttle from the zinc, or will be displaced and shuttle over to the aeroboronic acid, forming 2 boronic ester functional groups. This will effectively increase the fluorescence response in addition, or in proportion to the amount of pyrophosphate added. And from there, we hope to use this as a selective sensor for pyrophosphate. So now we'll go on to the experimental section. So, of course, first from the commercially available starting material, the synthesis of compound 2. The reaction went to completion with a near quantitative yield, or complete conversion to the product 2, and then NMR, nuclear magnetic resonance spectroscopy of the crude material showed the formation of the product within that mixture, which was very beneficial. Secondly, we protected with tert-butyl dimethyl chloride, or the TBSCl group, under SN2 reaction conditions. To do this, we first deprotonated the carbamate group with the base imidazole, and then upon that nitrogen lone pair will attack the chlorine, displacing it and forming that silicon-oxygen bond to effectively protect the alcohol. And of course, we got a yield of 3.4038 grams, or 84% yield. From there, we did a second protection with, again, using N-butyllithium to deprotonate the carbamate group. This is an extremely strong base in which is an organometallic where the normal butyl group will deprotonate the Bach group, and then because this is an extremely weak base or weak acid. Upon that, the lone pair on the nitrogen will attack the second Bach anhydride group and effectively dual protect the amine. And of course, we yielded 4.518 grams and a 97% yield. The next reaction utilized tert-butyl ammonium fluoride to effectively remove the TBS group. To do this, the fluoride anion in the group will selectively bind to silicon, which displaces freeing up the oxygen. Upon workup, it recovers the final alcohol. And we had a yield of 50, and then over these four protection and deprotection reaction steps, we had a yield of 53%, which this yield was 55%, which caused the major loss of product. The most reasonable explanation of this was that these four steps, or these four reactions, didn't involve a workup, so any starting material most likely skewed the results. So most likely our yield is higher than 53%, but because without purifying the crude, it affected the overall percent yield calculations. So here we have the nuclear, or the NMR spectra of our final alcohol, which is dual protected with here's the Bach group. As we see, the NMR uses a parts per million scale in frequency to determine the resonance effects of atoms flipping electronic states, which is very sensitive for which for this instance, we are using hydrogen NMR, which will be selective for different electronic regions of a molecule of those where those hydrogens are in the molecule. So for instance, lowest we have 18 hydrogens representing the 6 Bach or methyl groups on both the Bach groups. Due to these compounds being symmetrical, they will appear at 1 parts per million, or they will appear at the same scale in the parts per million. And of course, we had the integration yields 18 hydrogens for those 6 methyl groups. Secondly, we had the remainder of our hydrogens were present from the alkyl chain group, methylene groups. So we also don't see the alcohol. These often don't show up in NMR due to interactions with solvent, effectively exchanging between a deuterated solvent. So a lot of times the hydrogen is gone. So we didn't see that here. Continuing from this, we used, again, pyridinium chlorochromate, was used to selectively oxidize the primary alcohol to aldehyde. In previous research, we had attempted to only use a primary protected amine, but when using PCC, that yielded undesirable yields. So unfortunately, that is why we had to do several more steps to diprotect this Bach group, or diprotect the amine with 2 Bach groups. Again, this reaction undergoes a quick yield. It is quite near quantitative, and reaction due to the polarity of the starting material with the crude reaction mixture. A simple filtration through a plug of silica yields the final product, in our case 1.424 grams and 82.5%. Secondly, we have an excellent NMR of our yield. Again, we see near the low end of the spectrum, we have an integration of 18 hydrogens, again representing the 6 methyl groups, with each methyl group having 3 hydrogens. We then see 2, 4, 6 hydrogens related to the alkyl chain groups in between the spacing of the aldehyde with the amine. And also we see the presence of the aldehyde hydrogen, which is always seen around 9 to 10 ppm. So this was highly indicative of our purified aldehyde product. Peaks around 7.25 are always from the deuterated CdCl3 solvent, which interacts. Next, we performed the reductive amination step to produce compound 7. At the commencement of this semester, we did this reaction on a small scale to practice the reaction. Reductive aminations are often notoriously difficult. They can be reversed with water, so removal of water is one of the main driving forces of these reactions, and maintaining anhydrous conditions while also selectively reducing the imine product over reducing the aldehyde creates often low yields. So with this test reaction, we were able to yield 33%. Unfortunately, our automatic column that we were running to purify had a slight issue where it was separating the chromatography peaks. Usually one compound will elute as one peak on a separatory column. There are several issues, but that's beyond the scope of this presentation. Next, we scaled up the synthesis of compound 7 using the aldehyde that I synthesized this semester. Again, from previous semesters, the dipicoylamine containing mode I produced spring of 2020. And the mass of the crude that we collected was 1.5143 grams. Again, purified in the automatic column. We had to do 2 purifications just because of the amount. With the size of the columns that we run, the max is around 800 milligrams. So 2 purifications were required. Overall, it yielded 0.6264 grams with a 44% yield. That was relatively increased over the small scale reaction. So here we see the NMR of our reductive amination product from the first purification of the scaled up reaction using the produced aldehyde. Over here on the high end of the spectrum, usually around 7 to 8, we see the aromatic region, in which we have 8 total hydrogens representing the symmetry between 4 distinct hydrogens on this aromatic ring. So those represent the picoyl group, the aromatic hydrogens on the picoyl. Looking on this, we see 2 CH2 groups which are high in shift because they're right next to the aromatic region. The aromatic region draws electron density into it. Also, the nitrogen does, which induces these 2 methylene groups to come at the same chemical shift and be relatively high at 4. Next, we have several distorted. The remainder of the CH2 groups are often long chain alkyl chains are distorted overall in the NMR. So those are what we saw here. And finally, we had the 6 methyl group, or the 18 hydrogens from the 6 methyl groups on the block. So this showed our pure product was actually formed. Unfortunately, some of these that integrate not to 1 were because the product retained a lot of solvent. So further drying was needed to get the pure product, but these were distinct of ethyl acetate. So we were sure that these were just solvent peaks and the reaction was actually pure. The final synthetic step is an SN2 displacement of the bromine on the commercially available 2-bromomethyl phenylboronic acid, which we used Hunning's base or diisopropyl ethylamine in polar acetonitrile. This effectively deprotonated the hydrogen on the secondary amine, which attacked the carbon displacing the bromine and forming the new nitrogen carbon bond. Unfortunately, due to time restraints, we are currently purifying, or pardon me, after that has taken place, the acetonitrile solvent is evaporated and DCM and trifluoroacetic acid is added. This effectively deprotects or removes both bond groups, freeing up the primary amine again. From there, my apologies again, we are currently finalizing the purification on high performance liquid chromatography. We had a slight setback this week to finalize this because the column needed cleaned from use in other runs. High performance liquid chromatography is required because this molecule is so polar that you use a reverse phase of silica in which the silica is coated with nonpolar carbon chains. This adheres, therefore nonpolar molecules adhere better than polar, and with this we are able to effectively purify the key intermediate 8. We will be finalizing this upcoming final week and weeks of the spring 21 semester. So overall, I would say that the spring semester was highly productive. We were able to characterize and I was able to characterize and purify and synthesize 7 of the compounds, including this key intermediate 8. Another, as I've just discussed, HPLC is vital for the purification of compound 8. The primary amine, the 2 picoyl groups, and the phenylboronic acid are all extremely polar, and if you attempted to purify this on normal silica in which hydroxyl groups are present, you would get no yield, or you would get low to 0 yield and lose this vital compound. Pardon me. And also, we've learned that through that reductive amination step gives poor yields of 44%. This is bizarre, as for the most part, aldehydes reacting with primary amines should produce higher yields. They're fast reacting and selective. This could have been several issues, but we will be working to improve this in further research. And finally, we are now 1 step away from our final proposed sensor. So from there, we will continue our effort of research in future semesters. And to continue with that, with intermediate 8, we're again 1 step away. The only thing we are required to do is add on a catechol containing fluorophore. And then this is usually done with an acyl chloride, which the amine will react quickly because acyl chlorides are fairly unstable, will react quickly to yield the final sensor. Again, HPLC will be used to purify this, as it's even more polar with the addition of several polar functional groups of the fluorophore. Likewise, we hope to scale up synthesis 8 for quicker reactions, or for more continued research. With this key, the reason intermediate 8 is key is because we can then add in a variety of fluorophores as long as they contain catechol, the catechol functional group here. With this, we are able to change the conformation of the final PPI containing analyte and may produce a more accurate and better fluorescence response, which is beneficial for the overall goal. So throughout the semester, I've learned many valuable skills that I hope to take into further research and into my career as a future. I hope to look to graduate school to continue with organic synthesis, so these are all wonderful skills to have. Some of these skills include safe reagent handling techniques, for instance with N-butyllithium, which is highly flammable if exposed and not handled correctly. Dry solvent preparation, for instance, several of the reactions, including the one with N-butyllithium, used tetrahydrofuran, which is notorious for radical degradation and requires drying and distillation to purify for use in reactions. And of course, proper reaction setup in a variety of scenarios, setting up argon atmospheres, setting up glassware for effective reactions, handling of glassware, or all real world applicable skills that I've learned. Likewise, after the reaction is complete, reaction mixture workup using separatory techniques and product isolation using manual and automatic columns were gained and are highly valuable in the field that I wish to continue with. Likewise, Once the purified compound had been collected, methods for product characterization, including the chromatography mentioned, nuclear magnetic resonance spectroscopy mentioned, and mass spectrometry was successfully performed. These allow us to selectively identify and characterize the compound as the correct molecule and continue our research from there. And again, expanding upon my knowledge from Organic Chemistry 1 and 2 courses last year, an overall increased knowledge in organic reaction mechanisms, standard reaction procedures, and safety in the laboratory were gained. Alongside of this, I was able to learn what it really was to be a full-time researcher. While I wasn't performing real-time, being able to learn important time management skills, focus in the laboratory, and these were useful to effectively maintain my time both as a student and as a researcher, and it was really wonderful, a really wonderful experience to be able to use these in lab and see what I need to focus on and what will be beneficial for me in the future as I continue with my studies and future careers ahead. And finally, I'd like to thank my faculty mentor, Dr. Pavel Anzenbacher, PhD students Andrei Penovic and also Rajevic, who aided me throughout this entire process. And likewise, the funding for this project from the CURS program was gratefully acknowledged and gratefully appreciated. So that is the end of my presentation. I thank you all for participating and taking a look, and I wish you all the best as you continue on enjoying the symposium.